Hello and welcome students. In this video, we are going to take a look at the topic of tonicity and basically in relation to the plasma membrane and how the metabolic functions are affected by the tonicity of the solution. So we'll start with the very first note-taking point, that is the basic definition of tonicity. And basically what it means is it denotes the relative solute concentrations of two environments separated by a semi-permeable membrane and as you know the plasma membrane is semi-permeable. So let us take a look at what it basically means. And in the middle, let us say we have a semi-permeable membrane right here. And this area has a lower tonicity and this one has a higher tonicity. And by that, what does it actually mean? Let us say this side has 25% of sodium chloride and then 75% of water, as compared to the other side, which has 75% of sodium chloride and only 25% of water. So hope it makes some sense that the solute concentration is lower in this area and it's higher on this side. And the semi-permeable membrane is right here in the middle, which is going to actually allow the permeation or the movement of the materials through the membrane. All right, let's move to the next page, where we are going to take a look at three types of solutions based on tonicity. So they are categorized or classified into three forms, three types, and those three types are hypertonic, isotonic, and hypotonic. Now for hypertonic, you need to remember higher, greater than symbol. Isotonic, equal to symbol will help you remember. And then hypotonic below, less than symbol. All right, let us take a look at their definitions now. And before we start with the actual definition or what it means, with a picture or a diagram, things will be crystal clear to you. So let us draw an example of solutions here. And we are showing the cell right in the middle. All right, let's start labeling our cell now. Here inside I have 0.1 NaCl, 0.5 Here I have the concentration 0.1 of NaCl, and here I have also 0.1. And as I'm labeling, I think it will be making more sense to you with the names that have been classified. All right, so here basically what happens is that the solute concentration of the solution is greater than the solute concentration inside the cell. And as you can see that, that is what is happening. 0.5 is greater than 0.1. All right, here the solute concentration is equal to solute concentration of the solution is equal to the solute concentration as you can see they are similar here. All right, the solute concentration in the solution is going to be less than the solute concentration inside the cell. And that is what is happening if we take a look at the numbers. All right, so let us check what it means. Basically, solution is greater than the cell. Here, solution and cell are the same. Here, solution is less than the cell. All right, so that was about the three types of tonicities. And let's move on to the type of water movement that happens in these three different types on the next page. All right, so we will start with the very first one, which is our hypertonic solution. So here, remember our solution is greater than. So, there is a way to remember like hyper, when something is hyper or higher, than the regular amount or inside the cell. So what basically happens is we'll draw a diagram explaining it. So always remember when we have high solute, when the solute is higher, then the water is going to be low because total is 100%. If the solute amount is higher, then the water is going to be lower. And the opposite will be if the solute is lower, then the water is going to be high. So that is something to remember also. What will happen is we'll have, let's show the transportation using arrows. So we already, that means the Solution has a higher than the cell, so the water inside the solution will be less than the cell. So I hope it makes some sense to you that the water concentration is lower and solute is higher. So the opposite happens. So what basically happens in these type of solutions is so the movement of water molecules will be outside the cell. Because, by the definition of passive transport, they are going to move from a low concentration to a higher concentration. All right, let's move on to the next one, isotonic. Now, when the water molecules move outside the cell is going to shrink. Now, let us take a look at isotonic. In the isotonic solution, basically what we have is it's similar. It is equal to, so it's going to be 0.1 is the solute concentration, and it's 0.1 outside the cell also. So what will happen is an equal amount will go to the cell and pretty much the same amount comes out of the cell. Solution and cell are the same. Hence, the water concentration is also the same in solution and cell. So the water is going to move inward and outward. So the cell will maintain its shape. All right, so here the cell is going to shrink. Here the cell is maintaining its shape. On the next page, we are going to take a look at hypotonic. Solutions. With the hypotonic ones, what happens is basically, let's again show a diagram. And here we have very low concentration. So low concentration, remember, of solute means very high concentration of water. So what will happen is that water is going to travel to the cell. And here, high concentration of solute means low of water. So solution's concentration of solute is less than that of cell. Hence, water is greater in the solution as compared to cell's water. So what will happen here? Water will flow from the solution to the cell. The cell continues to swell. and may burst, causing cytolysis. All right, on the next page, we are going to take a look at tonicity in living beings and how it's related to animal and plant cells. And the very first point of note-taking will be osmosis happens. Now, plant and animal cells act differently, and that's what we will see. When water moves in and out. All right. Next point. You see, basically what happens is that plant cells are at an advantage. They have a cell wall, which actually helps to retain their shape. So that's where they have an advantage as compared to animal cells. All right, so again, we will make a T-chart here where we are going to have a comparison going. My lines are not very straight, but all right, again, the same thing, hypertonic, and how it changes the cells. Isotonic, where we have equal, and then hypotonic. And here we are going to look at animal cell and the plant cell. All right, so this one is going to be shriveled up cell because the water is moving out. So the cell is going to be looking like a shrinking. And here, because we have the cell wall, it's gonna look a little different. All right, here, same equal amount H2O goes into the cell, leaves the cell. Not a lot of difference. And then here, water is just entering from everywhere. So it's like a swollen cell. All right, so that is what it basically means with the diagram approach. Let's now take a look at other note-taking points. Let me check what point. Point number 3, all right? So we have point number 4. Plant cells like to keep the water in. To make them strong and support metabolism. Next, now basically what happens, there is a pressure that's exerted on the cell wall. And this pressure that is exerted in plant cells on the cell wall is called, I hope I don't butcher it, Turger pressure. All right, next up, applications. Saltwater fish pump out salt from their specialized gills so they do not dehydrate. Another application, when IV is given to the patients, they need to make sure that the cells do not explode or shrink, because water is going to travel or being transported from the cells or to the cells. So tonicity plays a major role, in living beings, and we looked at 3 different types of solutions that are based on tonicity: hypertonic, isotonic, and hypotonic. So I hope it made some sense. This video was pretty long, so I will let you go right now. And thank you for watching and adding more, views to my channel. I appreciate it. I'll see you in my next video, students. Bye.